Okay, sorry about that. My surface crashed. Okay, so we were saying CHO because carbohydrates have carbon, hydrogen, and oxygen. And then lipids are also CHO, right? Carbon, hydrogen, and just a few oxygen. Proteins are CHON because they have carbon, hydrogen, oxygen, and nitrogen. And nucleic acids are CHOMP because they have carbon, hydrogen, oxygen, nitrogen, and phosphorus. Okay? So let's talk about the monomers. Let's review the monomers for these categories. Okay? So, in order for life to have arisen, we have to go from monomers to polymers. The monomers of carbohydrates are monosaccharides, right? The monomers of lipids are fatty acids, right? We have the fatty acids and we have the glycerol, and we make either phospholipids or triglycerides. Proteins, we have amino acids that have the R group, remember? And we have our amino group and our carboxyl group. And then last are nucleic acids. There's an H there, yeah. And the monomers of nucleic acids are nucleotides. And these guys have the carbohydrate, the phosphate, and the nitrogenous base. Remember all that stuff, right? Our monosaccharides are 4 or 5 or 6 sided rings. Now, we can't just use monomers, right? We know that cells aren't composed of monomers. They're composed out of polymers. So carbon compounds had to be able to assemble into polymers, which contribute to cellular structures. So the polymer of monosaccharides are polysaccharides, right? The polymer of lipids are phospholipids, cholesterol, steroids. The polymer for amino acids are polypeptide chains, and then you put those together to form proteins. And then the polymers of nucleic acids are DNA and RNA, right? And now each of these are a part of different cellular components, right? So we have the cytoskeleton and the cell wall that are produced by carbohydrates, are composed of carbohydrates. We've got cell membranes and vesicles, or if you're Albert, vesicles, and communication, right? Cell communication. We'll talk more about that later. And then proteins are used in cell membranes. We have those transmembrane proteins. We also have a lot of enzymes here, right? So enzymes. So these are really important to different cellular functions. They're not their own structure, but they're super important. And then we have cellular structure, DNA and RNA, right? They're important. So the reason we did topic 2 instead of topic 1 first is so that you could be familiar with all this. Now remember, all this stuff, all the carbon and hydrogen and oxygen, we need to find out where these existed in early life and figure out how they formed monomers. And then we know that given the right conditions, these monomers will self-assemble into polymers. Now, the next question is, how do we go from these polymers to cellular structures? Okay. In order to follow that path, in order for life to have emerged, there are a bunch of different things that needed to happen. So this is understanding 2 still. The first cells must have arisen from non-living material. So here we have primitive Earth, right? We've got really warm temperatures. It's hot. We've got lots of radiation from the sun. The ozone layer hasn't really formed yet. We've got a reductive atmosphere. Right? We have lots of CO2. We don't have a lot of oxygen though. So chemical evolution happens first. So as the Earth cooled, we have the formation of inorganic chemicals. Inorganic chemicals have the ability to capture energy. That energy capture results in small organic molecules. So if you have inorganic molecules and you add energy, like from the sun, right, you can automatically start to see the creation of small organic molecules. Now, we can actually replicate this in the lab through a series of experiments. They're called the Miller-Urey experiments. So we have scientific evidence. Actually, we have scientific evidence that this happens too, right? So we have evidence for these two steps. Now, these small organic molecules have to get bigger, right? These are basically our monomers right here. And then they undergo the process of polymerization. Now, we're not talking like Yu-Gi-Oh! Polymerization, although it's a great card. We're talking about going from our monomers to polymers, right? So we get our macromolecules, lipids, right? So carbs, lipids, nucleic acids, and proteins. And we also form the plasma membrane. So we know that if you stick a bunch of phospholipids in water, that they'll automatically form that membrane structure. So everything up until this point, these are all chemical processes. You'll see chemical evolution. No life required. We know that these things can happen on their own given the correct conditions. And then from this point, now we start to talk about biological evolution, right? So you get your first protocell, which eventually can we would consider a cell, so like a prokaryote. It's important to understand the difference between chemical evolution and biological evolution. Chemical evolution has to happen first. We have to get the organization of these macromolecules into structures, and then we see the formation of life. And of course, we know that cells use predictable and repeatable chemical processes to live. Photosynthesis to store energy, right? And cellular respiration to use it. Now, these are life, right? So this is the point at which, when you think of life, that's what we're talking about. So let's talk about some of that evidence that I said we had. So we're talking about, oh, I should erase all this stuff. Can I erase all this stuff? Okay. So we're going to talk about how we go from here to here. And this is Miller-Urey. These are the experiments, the Miller-Urey experiments, okay? So here we are. Oh, it's also apparent in Haldane. Forgot about them. I always forget about them. So this is application 2. The Miller-Urey experiment and subsequent experiments demonstrate that it is possible to form all 20 amino acids commonly found in living things, nucleic acids, and some sugars, lipids, and ATP in early Earth-like conditions. So their experiments are showing evidence that this part can occur.
Stated Clearly presents, what was the Miller-Urey experiment? It was once believed that if you left food out to rot, living creatures like maggots and even rats would simply poof into existence. The idea was called spontaneous generation. A series of experiments starting in the 1600s disproved this idea, and in the 1800s, a new scientific law was proposed. Life only comes from life. It's true that rats, maggots, and even microbes are far too complex to simply poof into existence, but in 1859, English naturalist Charles Darwin put forth the theory of evolution. In it, he showed that under the right circumstances, relatively simple creatures can gradually give rise to more complex creatures. Given this information, serious thinkers began to wonder, is it possible that simple life forms actually could come from non-living matter? Not by poofing into existence, but through a natural gradual process similar to what we see in biological evolution. Darwin himself mentioned this idea when writing to a friend. But if, and oh what a big if, he wrote, we could conceive in some warm little pond with all sorts of ammonia and phosphoric salts, light, heat, electricity, and so on present, that a protein compound was chemically formed, ready to undergo still more complex changes. In 1924, Russian biochemist Alexander Oparin published a book which he titled The Origin of Life. In it, he outlined his thoughts on a gradual progression from simple chemistry to living cells. He imagined the early ocean as a primordial soup, a rich collection of complex molecules produced by natural chemical reactions. In this soup, further reactions could take place, eventually producing living cells. At the time, Darwin's warm little pond and Oparin's primordial soup were really just speculation. They were founded on a good understanding of chemistry and biology, but they could not be considered legitimate scientific hypotheses because no one had found a way to test or observe them. Science, after all, is the study of observable facts and an ongoing conversation about how those facts can be best linked together. Chemical reactions like those proposed by Darwin and Oparin are not expected to leave an observable fossil record. Without either having fossils to examine or a time machine to travel back and observe what happened, how could scientists even begin to study the origin of life? In the 1950s, Stanley Miller, then a graduate student at the University of Chicago, came up with an idea. We could simulate early Earth conditions in the lab and then carefully watch what happens. If you can't study fish in the sea, set up an aquarium. Working with his professor, Harold Urey, Miller designed an apparatus to simulate the ancient water cycle. Together they put in water to model the ancient ocean. It was gently boiled to mimic evaporation. Along with water vapor, for gases of the atmosphere they chose methane, hydrogen, and ammonia. These are simple gases which scientists at the time thought were probably abundant on the ancient Earth. They added a condenser to cool the atmosphere, allowing water molecules to form drops and fall back into their ocean like rain. The ancient Earth would have had many sources of energy, sunlight, geothermal heat, and even thunderstorms, so they added sparks to the atmosphere to simulate lightning. The goal of the experiment was not to create life, but to simply test the first step in Oparin's model. Can simple chemistry naturally give rise to the complex molecules of life? After running the experiment for just 1 week, their ocean became brownish black. Careful analysis revealed that through a series of reactions, many complex molecules had been produced. Among these were amino acids, special molecules of life that we once thought could only be built inside the bodies of living creatures. This was a pivotal breakthrough in science, so significant in fact that it gave rise to an entirely new field of research now known as prebiotic chemistry. Scientists don't know for sure if the gases used by Miller really were the most common gases of the ancient Earth. Because of this, many experiments have since been done, showing that the molecules of life can form in a wide variety of environments with different starting chemicals and different sources of energy. Sugars, lipids, and amino acids have even been found on meteorites. This suggests that the molecules of life formed all throughout the ancient solar system and may be forming right now in other regions of our galaxy. Together, these discoveries tell us that Oparin's primordial soup and Darwin's warm little pond could have easily existed in one way or another on our ancient planet. So to sum things up, what was the Miller-Urey experiment? The Miller-Urey experiment was our first attempt at simulating ancient Earth conditions, in this case, the ancient Earth's water cycle, for the purpose of testing ideas about the origin of life. The Miller-Urey experiment is significant for 2 main reasons. First, though it was not a perfect simulation of the early Earth, it clearly demonstrated for the first time that biomolecules can form under ancient Earth-like conditions. Second, the experiment took what was once mere speculation, the idea that life may have emerged from chemistry, and transformed a portion of that speculation into legitimate, testable science. Many questions remain to be answered about the origin of life, but scientists from many nations and many fields of study are now following Stanley Miller's lead. They're finding ways to turn those questions about the origin of life into testable, scientific hypotheses. Simulation experiments cannot tell us exactly how life formed in the past, but if enough of them are done, they could eventually tell us if it's possible for life to emerge from chemistry. I'm John Perry, And that's the Miller-Urey experiment stated clearly. This video...
Okay, so that video does a really nice job of simplifying what the Urey-Miller experiment was all about. We're going to continue talking about experiments pretty much from here on out for the rest of SL Bio, because you have to have a good understanding of the foundation of where these ideas come from. So we started today talking about Pasteur and how he disproved the idea of spontaneous generation by boiling flasks with broth in it and watching microbial growth grow in broth that was exposed to air. We just learned about Urey-Miller's experiment that demonstrated that complex organic molecules can form under early Earth-like conditions. So we have more evidence for the common origin of life on Earth. And that's based on the fact that all organisms use the same genetic code. We all speak the same genetic language. Whether you're an amoeba swimming around in a pond, whether you're a plant, whether you're a fungus, we all speak the same genetic language. You can take a gene, which is a piece of genetic material, from any organism on this planet and put it into another organism on this planet, and that organism's cells will be able to read and understand that genetic code. That's what genetically modified foods are, or genetically modified organisms. They contain pieces of DNA, or genes, functional units of DNA, from other organisms to help them achieve a certain skill or a certain trait. So here we have DNA. We have our base pairs, A and T and C and G, right? These are our nitrogen base pairs, or nucleotides. This is a nucleotide. Remember? COHNP. The carbon, the hydrogen, and the oxygen are in the backbone, and so is the phosphate. And then the nitrogen is in the amino acid. I'm sorry, it's in the nucleotide. Now, 3 base pairs, right? So these 3 base pairs, you can use these 3 base pairs to produce an amino acid. Now, I should probably be careful about how I say that. They don't produce an amino acid. They contain the information on how to build an amino acid. So 3 letters, A, T, G, right, will give you an amino acid. So A, 1, T, O. I am like not doing so well today because this is for RNA. Because you know how I know? Because it's got a U. So let's see. So say I have a piece of mRNA, and we'll talk about DNA synthesis and protein synthesis in another topic. But say I have some mRNA and it's AUG, right? So here's my A, there's my 1, U, G, right? So A, U, G, and we would get our amino acid. We also have evidence because many genes across different organisms are the same with only slight variations. So here you have a list of bacteria, and this is a series of part of the genetic sequence. And you'll notice these arrows, and the different colored arrows represents codes that are the same in the DNA of that organism. And you'll notice they all look pretty similar. Like there's some differences. Like see, for this guy, you see this big gray arrow. For this guy, you see it's broken up a little bit. So there are some changes. This guy doesn't have it. But for the most part, they're pretty similar. And that makes sense because no matter what you are, if you're a living thing on this planet, you're going to use the same chemical processes. You're going to use either aerobic or anaerobic respiration. If you're a plant, you're going to use photosynthesis. And these genes contain the instructions on how to build the proteins that make all these things work. So it makes sense that a lot of our genetic information across species on this planet are the same. Okay. Understanding number 3. The origin of eukaryotic cells can be explained by the endosymbiotic theory. So hopefully we all remember the difference between prokaryotic cells and eukaryotic cells, right? I thought there was another. I guess not. So prokaryotes came first, right? Prokaryotes are simple bacteria. Eukaryotes came later. Endosymbiotic theory explains how prokaryotic cells, so bacteria, right, became eukaryotic. Things like plants, animals, protists, fungi. The big difference is prokaryotes don't have a nucleus and they don't have cell membrane bound organelles, right? Where eukaryotes have lots of different organelles, different compartments that help separate conditions so that different chemical reactions can happen. Well, how did we go from cells, prokaryotes, that don't have chloroplast and mitochondria to eukaryotic cells that do have chloroplast and mitochondria? Because those two things, chloroplasts and mitochondria, they are the key to multicellular life on this planet. Without those guys, we'd all still be bacteria, right? What we think happened is that some prokaryotic cell, right, ingested a chloroplast, but instead of digesting it and breaking it down, it formed a symbiotic relationship. Now remember, a chloroplast can harvest energy, right? So now, if you're a prokaryote and you sucked up this chloroplast, you can either digest it and be done with it and keep going about your life, or if you maintain that chloroplast, you've got an energy producer for you located inside of you. That's a pretty beneficial deal. That would give you a serious competitive advantage over other prokaryotes. Now, the same thing probably happened with mitochondria. Remember, mitochondria break down glucose molecules or starch molecules or carbohydrates and produce ATP. But instead of anaerobic production, where you just get 2 ATP, right, glycolysis, you're going to get 36. That gives you a huge competitive advantage. You get all this extra energy compared to other prokaryotes that don't have them. This collectively is known as endosymbiotic theory, right? So endosymbiotic theory helps explain very specific characteristics of mitochondria and chloroplasts present in eukaryotes. Now, mitochondria and chloroplasts self-replicate like other cells. That's a good indication they used to be their own thing. They used to be their own prokaryotes. Both chloroplasts and mitochondria have their own DNA that is separate from yours. Right? So you inherit your mitochondria through your mother. It's maternal only. So mitochondrial DNA, that's how you can trace your maternal lineage. That's how we came up with the idea of a genetic Eve, right? So there is one woman that gave rise to all of the humans that are alive on our planet today. She's called mitochondrial Eve. We can trace all modern humanity back to one woman through mitochondrial DNA because mitochondria have their own DNA. Now, we also know that chloroplasts and ribosomes use a 70S ribosome instead of the 80S ribosome that you see in regular eukaryotes. So that means in your cells have 80S ribosomes. Your cells also have mitochondria. Inside that mitochondria, there are 70S ribosomes, which is a good indication that mitochondria used to be its own prokaryote. Now, they also have double membranes, meaning they were taken in by a vesicle via endocytosis, and then that vesicle membrane just stayed. So let's talk about that in a little more detail. Here's our prokaryotic cell, right? So the first cells were prokaryotic, single-celled bacteria. And then over time, some prokaryotes evolved into eukaryotic cells, protists, plants, animals, and fungi. So here's our prokaryotic cell, pretty simple, right? And then here's our eukaryotic cell with all these organelles that all do different chemical things. They all play their own chemical role in keeping cells and organisms alive. So we had to get from here to here, right? So no longer are we talking about chemical evolution. Now we're talking about biological evolution. So chloroplasts and mitochondria have characteristics that prokaryotes have. They have double membranes. So you can see in a chloroplast, here's the outer membrane, and then there's the inner membrane. You can kind of see here, the orange is the outside of the membrane, and you've got that double membrane on the inside, the red for the mitochondria. These guys replicate via binary fission. They replicate on their own. They have smaller 70S ribosomes, and they also have their own small circular strands of DNA that look like plasmids, which are the same shapes and forms of DNA that prokaryotes have. So, big story short, your mitochondria, they used to be their own organism. They used to be their own thing. They got sucked up by some prokaryote. That prokaryote then had this great advantage over everything else because it could create so much more ATP. That extra energy is what allowed multicellular life to evolve. Similarly, you have some prokaryotes that sucked up chloroplasts. Now they have the ability to harbor energy from sunlight, right? So they don't need to keep eating stuff. They can create the energy themselves. That gives them a huge competitive advantage. So here's another graphic that just explains endosymbiotic theory. Here you have a really simple ancestral prokaryote minding its own business, right? You've got some folding of the plasma membrane for some basic organelle structures, like the endoplasmic reticulum, right? You got a nucleus starting to form, you got the endomembrane system. Now, at some point, that prokaryote engulfed a chloroplast. Oh, this is actually mitochondria, right? Mitochondria, which is great because now we can produce lots more ATP. And then eventually, some offspring of these guys also ingested a chloroplast. Right? And these guys later evolved into plants. And these guys evolved into animals and fungus and protists. I think the plants got the better deal, honestly. Okay, let's summarize.
This is you, and these are your ancestors, a huge pyramid stretching into the past and balancing right on your head. How many ancestors do you have? Well, you have 2 parents, 4 grandparents, and 8 great-grandparents. 4 generations back, your direct ancestors total 30. If we continue down this line, doubling every step, just 40 generations ago, we'd find 1 trillion ancestors all living at the same time, which is ridiculous. That's not only more people than have ever been alive, it's more stars than are in the Milky Way. Since our species came on the scene 200,000 years ago, there have been maybe 7,000 or 8,000 generations of humans leading up to you. So where are all your missing ancestors? Clearly there's been some inbreeding. We're not talking banjo-playing King of Spain Cersei Jaime inbreeding, but every family tree inevitably grows forks. Before Tinder, choices for mates were often limited to as far as you could walk. Even people like Charles Darwin and Albert Einstein married their first cousins. Because so many people with shared ancestors have reproduced, our number of actual ancestors is much smaller than what simple math tells us. If we replace that with fancy math, factoring in how people moved and lived and paired up, life expectancy, trade, geography, Genghis Khan, we find something interesting. Every human alive today shares a common ancestor in their family tree, and this person lived only around 3,000 years ago. That's right, next time you get in a fight with a stranger on the internet, just remember that you share the same great-great-great-great-great-great-grandfather or grandmother. But we don't know who that person was. The math tells us they must have existed, but they didn't leave fossils or artifacts or like a note or something. Though writing birthday cards for each of your 7.4 billion great great-grandchildren would have been a nice gesture, but we all carry a record of our ancestors in our genes because DNA is copied over and over. Every so often a mistake is written in. You know how when you make a copy of a copy, it doesn't come out as sharp? Like that. But since most of our DNA can be changed without affecting how things work, many of these mutations slip through to the next generation. These genetic changes accumulate at a steady rate through time, so scientists can read them like a molecular clock and estimate how much time has passed. And which changes individuals share tell us how closely or distantly related they are. Humans seem really different, but on a DNA level, we're remarkably similar. Groups of chimps in Central Africa living right next to each other show more genetic variation than we find in the entire human population. Now, this genetic similarity tells us that our species is new in the big scheme of things, and that at one point our population was small, maybe as few as 10,000 of us. To put that in perspective, that's only a third of your average Bruce Springsteen crowd. Sorry, boss. Today, any two humans only differ by about 1 out of 1,000 DNA base pairs. But our genome is so big, that's still millions of single letter differences, or SNPs, for single nucleotide polymorphism. We tend to see combinations of these changes, chunks of SNPs, associated with different geographic locations. Now, companies that test your DNA ancestry, they read thousands of these single letter changes in your genome to make a sort of signature of your unique genetic variation. Then they compare your signature to thousands of reference individuals from various parts of the world, and do a bunch of fancy math to see which parts of your genome most likely came from certain geographic areas. My genetic results pretty much look like this. My ancestors on both sides of my family are from Northern Europe and Scandinavia, which explains my last name, why I'm tall, why I don't tan, and also why I carry more Neanderthal DNA than 2/3 of people. If you're confused why I have Neanderthal DNA, you should watch our last video. I didn't find any surprises, but many people learn about ancestry they didn't know they had. Where we come from isn't always obvious on the outside, but DNA doesn't lie. Before using math, we identified an ancestor not too long ago that's related to all of us. But that person's genetic influence has been shuffled so much it's invisible in our DNA today. Is there someone whose genes have been passed on, unbroken to today? Some leftover fingerprint from the mother of everyone alive? There is. You have a 47th chromosome. It lives in mitochondria.
The powerhouse of the cell.
Okay, so we're doing that again. Mitochondria used to be free swimming. They have their own genetic material. Unlike your other 46 chromosomes, there's no shuffling when it's passed between generations. And what's more, all your mitochondria came from your mother's egg, not your father's sperm. They trace an unbroken line of ancestors stretching back through every female in your family tree. By comparing the changes that have accumulated over millennia, we find the most ancient human mitochondrial DNA comes from Africa, where our species originated. We can even trace it back to one woman, about 150,000 years ago. Other Homo sapiens females lived alongside her, but only her lineage lives on today. All other Homo sapiens lineages are extinct. This is mitochondrial Eve, and every single one of us descend from her. In the truest sense, we really are family, even if we're just 100th cousins or something. But our ancestry isn't just branches stretching into the past. It's also a tree that extends into the future. Today we have more power to mold that future down to the genetic level than we've ever had before. So what might our species' future look like? Next time. Stay curious. This video is part of a special series we're doing about the story of our species, where we came from, how we're all connected, and where we're going. If you haven't already, check out part 1 and 2 to trace the fossils in our family tree and learn why we're the only humans left. And be sure to subscribe so you don't miss any of our videos.
Okay. So that was a kind of a long video, but the purpose of that video was to provide some additional evidence about how all life on Earth is related, particularly in humans, right? We have evidence of endosymbiotic theory, of mitochondria and chloroplasts being their own prokaryotes and getting absorbed into a cell that later became eukaryotes. We have records of mitochondrial DNA in humans that go to show all humans evolved from certain common ancestors. Now remember, we started this lesson talking about how we went from chemical life or chemical chemicals to biological life. We have two different processes. We have chemical evolution first, which is the combination of elements under proper conditions to give rise to monomers and then give rise to polymers. Biological evolution doesn't take over until you have the formation of the first cell. Pasteur's experiments demonstrate that life comes from life. He disproves spontaneous generation, right? We had Miller and Urey's experiment that demonstrates that given the proper conditions, certain chemical elements can self-organize into the components that build modern-day cells. So at this point, you should be able to understand and answer these review questions. The first one is outline the structures necessary for the first cell to have emerged, right? And you can use this to help you. You should be able to outline Pasteur's experiment and explain how it provides evidence against spontaneous generation. You should be able to describe endosymbiotic theory and how it explains the emergence of eukaryotes. And you should outline evidence that supports endosymbiotic theory. Now, I'll also put these links in the page for you. These are the videos that we watched today, so if you need to rewatch them, you can. All right? All right, guys. Have fun. Remember, next rotation is your skills test on 1.1, 1.2, and 1.5. Make sure your notes are printed out so you can use them in class. Okay?